Good evening, everyone. This is Dr. Satya Yadav, and on behalf of Society for Pediatric Cell Therapy and Transplant, I invite you to another interesting webinar. This is the ninth in the series of webinars we have held so far, and all of them have been wonderful webinars covering such extensive topics in transplant and cell therapy. So today we have another interesting topic lined up, and today's session is dedicated to something which we all in our medical college, medical school days thought it's very futuristic and is going to happen, but now it's all happening. So gene therapy is very much here and it's very much happening. So today's webinar is focused on that. And for this webinar, we have a top-notch chairperson, Dr. Suparno Chakravarti, who is head of bone marrow transplant and hematology at Dharamshila Cancer Institute. And he's also honorary professor at MIT Institute of Stem Cell and Molecular Research. So, Dr. Suparno, thank you for being a chairperson tonight. And please take over and introduce the speaker. Thank you.
Yeah, thank you, Satya. Thanks very much for the intro. And your organization, or rather our organization, is doing a fantastic job, over the past few weeks and months in organizing these sessions, which are really enlightening. And today, I believe and I sincerely hope that all the young people in the field of pediatrics and transplant and cellular therapy would get to a world of sort of unknown, I would say, for people who are entirely in the clinical field. Um, we all hear about it a lot, but the nitty-gritty, and as it is called, the nuts and bolts of gene therapy, we have Dr. Siva Prakash Ramalingam with us today, who is um, an associate professor in the Department of Biological Sciences at IIT Kanpur. Um, he's got an extensive experience on genomic work, including targeting and going into the field of therapeutics from there, starting from his work at Johns Hopkins lab onto going from IGIV to IIT Kanpur, where he's doing both basic translational as well as therapeutic work with regards to gene therapy. Uh, so without much ado, I will ask Dr. Ramalingam to take over from here and keep the crowd enthused, where we would expect a lot of, interrogations to come up at the end of the talk, followed by a panel session, which should be equally interesting. Thank you. Over to you, Dr. Ramalingam.
Thank you. Thank you, Dr. Satya, both Satyas and Dr. Chakrabarti for the kind introduction. So without any delay, I'll just start. I'll just share my screen. Just yeah. So here you go. So hope you can see my screen. Can you just say yes if you can able to see my screen?
Sir.
Yes. Okay. Fantastic. So today, thanks again. I'm going to touch upon cell and gene therapy from introduction to biomedical applications. So I will start with very basic viral vectors. What is gene therapy? What is ex vivo gene therapy? What is in vivo gene therapy? And I will give you some applications which already has reached the clinic. And then what are the limitations in this cell and gene therapy, especially using the lentiviral or retroviral vectors. And then how to overcome that. So how to go around that one. So that's where the targeted genome engineering approach comes. So I will talk about the gene editing nucleases, which is where my expertise falls in. zinc finger nuclease, talons, and CRISPR, or the basics of the gene editing nucleases, and how what are the applications, and then I will give a few examples from HIV to what we are doing in my lab at IIT Kanpur for sickle cell anemia and thalassemia. So I just start from very basic. What is viral vectors? So viral vectors are the tools used to gene therapy to deliver. Say example, if you are coming to your hospital from your home, you use some vehicle, right? Either use car or two-wheeler or any other mode of transportation. So that vehicle here is the viral vectors. So it carries the genetic material and transports into the cells. Okay. So this virus have been engineered to make it more safe. So these are all the natural viruses. So this is being used, repurposed for delivering the genetic material into the cells. So there are multiple key features in this. So there are multiple key viral vectors. One is the retroviral vector, adenoviral vector, or lentiviral vectors, or adeno-associated viral vectors. So these are all very commonly used nowadays in gene delivery systems. So what are the applications? As I told, one is for the gene therapy, for treating various genetic disorders, or even for cell therapy like CAR T cell therapy. And even it has been used recently for delivering the COVID-19 vaccine, for example, AstraZeneca one, or the Indian Bharat Biotech ones are using these viral vectors. So yeah. So what is gene therapy? As I told, you say example, you have genetic disorders like sickle cell, thalassemia, hemophilia, or Gaucher's disease, or any genetic disorder. You have a faulty gene. Okay. So if you want to replace this faulty gene into a therapeutic gene or the wild type gene, you need to have some kind of vehicle to deliver your therapeutic gene into the target cell. Okay. So that's where you take these therapeutic gene, package it into a viral vector, and then you can deliver it into the human cells. So this has two types. One is ex vivo gene delivery, and then the in vivo gene delivery. Okay. So ex vivo means you have a therapeutic gene and then you have packaged it into a viral vector. Either it can be a lentiviral vector or retroviral vector. So in this case, you take out the stem cells, either the hematopoietic stem progenitor cells in the case of sickle cell disease or thalassemia, using the apheresis, and you can just deliver these viral vectors into the CD34 hematopoietic stem cells ex vivo, and then you can put it back into the patient. So this is very similar to your bone marrow transplantation. So in bone marrow transplantation, we always look for the donor, suitable donor, HLA matched donor. But here it is autologous. So you are taking patient's own cells and you are modifying it, say example, engineering it, and then putting it into same patient. So there won't be any immune rejection. Okay, that's the major advantage here. So these patients have to go for conditioning regimen, but if it is in the bone marrow transplantation, the conditioning is very severe, but here the conditioning can be milder. So since it is own cells, the engraftment will be much better than the donor cells. Okay, so there won't be any immune rejection here. The second approach is in vivo approach. So here mostly the adeno associated viral vectors are used. So you package the therapeutic gene and then put it into the viral vector, the adeno associated viral vector, and then directly you can intravenously inject into the patients. Okay. So the advantage of the adeno associated viral vectors, so it has a tropism. Okay. So it has different type of serotypes. Okay. That serotype has a specificity. Say example, adeno associated viral vector serotype 9, it specifically targets the neurons. Okay. So AAV6, it mainly targets the hematopoietic stem progenitor cells. So this has certain advantage in terms of the serotype. So the major advantage is you don't need to take out any patient cells outside, and then you have to modify this. So you can just make this adeno-associated viral vector particles in the GMP, good manufacturing condition, and then directly inject intravenously into the patient. So then this therapeutic gene goes into the cell, and then it gets translated into the mRNA, and then it produces the protein, and then it replaces the mutant gene or the pathologically relevant gene which causes the disease. Okay, so there are some examples I just give you here. So one is the SMA. It's spinal muscular atrophy, so which is a very rare disorder, the neurological disorder here. So here what people have done is they have taken a AAV9, as I told earlier, so packaged this SMN1, the survival motor neuron gene, into the AAV vector, and then they intravenously infused into the patients, and then since AAV can pass through the blood-brain barrier, it goes into the neurons, and then the gene gets started producing the mRNA and protein, and then the patient will it reverses the disease phenotype. So it's basically a one-time intravenous injection. So the transduction of motor neurons likely results in improved motor neuron development, function, and survival with potential near normal attainment of motor milestones. So this is one example. This was developed by Novartis, but still it is very expensive for Indian patients. It costs around more than 10 crores. Okay, so the next example is, okay, so the next example is, I have given an example for adeno-associated viral vector. So that is like an in vivo approach. The second approach is the lentiviral based approach, which is an ex vivo approach. Okay, so this is already been approved for sickle cell anemia and thalassemia, which was developed by Bluebird Bio. You may know that. So I'm just giving an example here for the transfusion dependent thalassemia patients. So what they have done here is they have taken a beta globin wild type gene, and then they packaged it into the lentiviral vectors along with the signaling molecule. So you need to have a LTR sequence which regulates the beta globin or any globin gene sequence. Okay, I will come to that, the regulatory sequence, in the second part of my talk, where I am going to talk about the genome editing. So here they have packaged the wild type gene with the signaling molecules like HS2, HS3 and HS4, which are the signaling molecules which is essential for beta globin synthesis in the erythroid cells. So they have taken the cells from the patients, and then they packaged it into the, they delivered through the lentiviral vectors, and then they put it back into the same patient. So it is like an autologous cell replacement therapy. Okay, so this has been successful, more than 95% successful in this case. Okay, so this is also the cost is very expensive. So that's why the many patients across the world couldn't able to afford this because of the $3 million price tag. Okay, so these technologies, though it works very well, I'm not going into a CAR T cell therapy. I think it has been already covered in one of the session. I heard that. So that is the cell therapy. So since it has been already covered, I am not going into the cell therapy. So I just covered only the gene therapy part. So there are some limitations also which has for this gene therapy approach for both AAV and the lentiviral approach. So the lentiviral approach has two major limitations. Okay, so one is in our cells actually. So the homologous recombination is the experiment. It happens 1 in 1 million cells under normal condition. Okay, so that's where these lentiviral particles go and integrate randomly in the genome, and then the genes get started expressing. Okay, so so that causes two major problems. Okay, so one is the non-targeted random integration. So that may lead to insertional mutagenesis and the oncogene activation. Say example, if you have the lentiviral particles which goes and integrates into a oncogene site. Okay, so then the oncogene will get activated, and then eventually it will lead to leukemia. Okay, so that is the that will lead to unintended effects. So That is a major limitation in this field for the retroviral vectors and the lentiviral vectors. So one of the best example is the SCID therapy. Okay, this happened in early 90s, 1990s. Uh, as you know, that SCID is a severe combined immunodeficiency, which is also called as a bubble syndrome, which is caused by a IL-2 receptor gamma gene mutation in the IL-2 receptor gamma gene. So what they have done here is they have taken this wild type gene IL-2 receptor gamma, and then they packaged it into a retroviral vector, and then they delivered it into the 10 patients in the phase 1 clinical trial. Okay, so very unfortunately, one kid developed a leukemia after infusion. Okay, so and then they went back and checked why this has happened, the leukemia so when they did the whole genome sequencing, and then they found out that these retroviral particles integrated in the oncogene site, and then oncogene got activated, and then that developed the leukemia. So that's where this miracle gene therapy trial has halted in 1990s and early 2000. Okay, so then the scientists were thinking about how to go around this one. So then now people are developing, the scientists have developed the third generation lentiviral and retroviral particles, so which is much more safer than the old generation retroviral or lentiviral particles. So that's where these recent generation lentiviral particles are being mostly used in the CAR T cell therapy for either ALL or DLBCL or multiple myeloma and multiple other diseases, even for sickle cell and thalassemia. So the random integration is the insertional mutagenesis and oncogene activation is very less in these recent generation lentiviral and retroviral vectors. So how to go around this random integration, how to stop this random integration or non-targeted insertion? So one approach is the targeted genome engineering or targeted gene editing. The question we and others ask, can we precisely edit or modify the gene or genome? Say example, if you have sickle cell mutation, if you want to change the mutant GTG into GAG, okay, so val into glutamic acid, or if you want to correct the beta thalassemia mutation, minus CTT, the deletion, if you want to correct that mutation, or there is a 619 base pair deletion in very common in Indian patients. So can we be able to correct that one? Or can be able to correct the Gaucher's disease mutation L44P, which is very common in Indian population. So is there a tool or the technology currently available to correct specifically to those mutations wherever it is present, instead of going randomly in the genome? Can we just targetedly we can able to correct that one? Answer is yes, using gene editing nucleases. Okay, so these are all I have listed some of the hereditary or genetic disorders. Now you can use gene editing nucleases to correct sickle cell mutation or thalassemia mutation or cystic fibrosis, the CFTR mutation, or muscular dystrophy, DMD gene mutations, and then spinal muscular atrophy, ataxia mutations as well. So you can use all these genetic disorders can be cured. The major advantage is this will achieve a curative. These are all the curative therapies, the cell and gene therapy using either zinc finger nuclease talents or target or CRISPR-Cas9. Okay, so this is the simple definition for genome editing. What is genome editing? Okay, genome editing is the process of introducing specific modifications in the endogenous DNA, including it can be a deletion or point mutation, inversions or translocations by making targeted double-stranded break. Okay, so all these gene editing nucleases simply makes targeted double-stranded break. Okay, so once the targeted double-stranded break happens, so the cells has its own repair mechanism. Okay, so there are two ways to repair. One, you can provide the extra chromosomal DNA with appropriate homology arms. I will come to that. And then once the double-stranded break happens, by homology directed repair, the mutation gets corrected. Okay, so what is the major advantage of the targeted double-stranded break is, so if you make as I told earlier, in a normal scenario, the homologous recombination happens 1 in 1 million cells, 1 in 10 lakh cells. Okay, but if you make a targeted double stranded break, you can increase the homology directed repair into several thousand fold. That's where the gene editing plays a major role in gene correction or making the gene knockouts. Okay, so that's where I have in the schematics, you can see here, under normal condition, this happens only 1 in 10 million cells. Okay, but if you make a targeted double stranded break using gene editing nuclease, CRISPR or other any nuclease, you can increase the homologous recombination, homology directed repair into several thousand fold. Okay, so that is where the major advantage here. Why genome editing? So it has one, yes, obviously it can be used for correcting the disease mutations, and then you can create a beneficial genetic mutation, say example in HIV. I will come to that. This is a biomedical application. Okay, these are all the curative therapies. It leads to the curative therapies. But in our human genome, we have around 20,000 genes. Okay, in these 20,000 genes, we know only the function of 6,000 genes. Okay, the remaining 14,000 genes, we don't know what is the function. Okay, whether it is essential for our cell growth and development, or these are all junks. So if you want to ask a basic research question, whether this gene X or Y or Z, whether it is essential for cell growth and development, you can do a in vitro disease. You can create those mutations in a relevant cellular model and then study the basic biology. Okay, whether this gene is essential or this gene is controlling some downstream pathways, or it is controlling upstream gene expression. All these questions can be answered using making this targeted double stranded break using gene editing nucleases. Okay, that is where the major advantage here. Okay, so there are three different generations of gene editing nucleases are there. So we are all very familiar with CRISPR-Cas9, but before CRISPR-Cas9, there are two other technologies which was already developed and reached clinic way back in 2009-10 itself. Okay, so the first gene editing nuclease is a zinc finger nuclease. Okay, zinc finger is a DNA binding protein, which is the most abundant DNA binding protein in the eukaryotes. Okay, including the human beings. Okay, so what happened is, Dr. Srinivasan Chandrasekharan from Johns Hopkins, what he has done is, so before 2000, there were no hybrid restriction enzyme or gene editing nucleases. Say example, if you want to make a specific gene knockout, targeted gene knockout or targeted gene correction, there was no technology in that time. Okay, so what he came up with the solution. He developed a gene editing nuclease, the first gene editing nuclease called zinc finger nuclease. So what he has done is, he took the zinc finger DNA binding protein. Zinc finger is a DNA binding protein, and then he fused a FOC1. FOC1 is a restriction enzyme. So we all might have studied the restriction enzyme type 2 and type 2S restriction enzymes. Type 2S restriction enzyme is this type 2S restriction enzyme has a two domains. One is DNA binding domain, another one is a nuclease domain. Okay, so he took this FOC1 nuclease and he separated these two things, and then even after separation, the FOC1 nuclease domain is still active, functionally active. So he took the zinc finger DNA binding protein and then the FOC1 nuclease, fused it and created a zinc finger nuclease, artificial gene editing nuclease. Okay, so zinc finger is a DNA binding protein, as I told, which has a beta sheet and a helical structure. Okay, so this helical structure, each finger can able to recognize 3 fingers. Okay, so you can able to fuse 6 or 7 fingers. So 6 fingers means 6 into 3, 18 base pair. It can able to recognize 18 base pair, and then the 3' end of the zinc finger DNA binding protein, you can put this FOC1 restriction enzyme. Okay, so the FOC1 in order to make the targeted double, in order to make a double-stranded break, you need to have a dimerize. You need to dimerize. Actually, you need to have a two molecules. Okay, dimer means it's two molecules. Okay, so you need to create left zinc finger and then the right zinc finger. Okay, so the left zinc finger can recognize 18 base pair, and then the right zinc finger can recognize another 18 base pair. Wherever you want to target, you can artificially synthesize that, okay, by using molecular biology techniques. I'm not going into those details in detail, it will take very long time. So this 36 base pair in the genome, okay, say example in sickle cell anemia, you want to target the beta globin gene. So you can design a zinc finger DNA binding protein and then fuse it with FOC1 nuclease. So that will target the 36 base pair at a stretch. Okay, that at a stretch of this 36 base pair may not be present elsewhere in the genome. That is where these gene editing nucleases brings the specificity. Okay, so if you make this the DNA, zinc finger nuclease DNA, and then if you deliver it into a cell of interest, say example in the sickle cell anemia or thalassemia, if you put it into CD34 hematopoietic stem cells, it goes into the cell and then it recognizes, it goes and binds to the beta globin gene wherever the homology is there, and then the FOC1 dimerizes and it makes the targeted double-stranded break. Okay, that's what the zinc finger nuclease. Okay, so this has some limitations. Each technology has some limitations, right? So if you are buying a iPhone or anything, any phone, so or any software, initially it comes and then later there are maybe lot of bugs, it will get fixed, right? So but this here also there are some limitations. Scientists are trying to fix those bugs. Okay, one major bug here is if you are fusing 4 fingers or 5 fingers, the binding is influenced by the neighboring ones. Okay, that is the major. If this finger binds, if this finger doesn't bind properly, it will influence the neighboring one. Okay, so that may lead to off target effect. Okay, that is where the major limitation in this. So in order to overcome that, scientists have come up with another solution. So this is John Poch from Max Planck Institute. He's a plant pathologist. He was working in a plant bacteria called Xanthomonas. Okay, so he found out another DNA binding protein which is called the tail transcription activator like effector protein, which is very similar to zinc finger protein. But only the DNA binding protein is different here. It is a tail here, there's zinc finger. So people already know that the zinc finger nuclease is existing. So what they have done is they took this tail DNA binding protein and then they fused the FOC1 same enzyme and then they created tail talons. It's a transcription activator like effector nucleases. So here the major difference is each tail domain recognizes only one DNA base pair. Okay, there each zinc finger domain recognizes 3 bases, but here it recognizes only one base. Okay, so that is the major advantage, and then it is not influenced by the neighboring tails. Okay, so it gives high specificity, and then you have left 20 base pair recognizing tail domain, and then right you have another 20 base pair, that 40 base pair at a stretch in the genome will not be present anywhere else. Okay, that's where the specificity comes. So this has very less off target effects, but it takes time to produce these things. Okay, so this is the talent. It looks like, the structure looks like this one only. You have the tail DNA binding domains. This is the top strand, and 5' to 3' strand and 3' to 5' strand. So if each tail domain recognizes single base pair, and then once it binds, the FOC1 is the nuclease enzyme. It makes the targeted double-stranded break. It cuts here. Okay, which is very similar to zinc finger nuclease. Okay, so then when this also reached clinic, and then there were multiple clinical trials were going on using zinc finger nuclease. Also there is a company called Selectis Bio, which is a French-based company. They are now doing off-the-shelf CAR T cell using these talents. Okay, there are papers, 2-3 papers came last year and 2-3 years back also. They are doing they are trying to develop allogenic CAR T cells using for hematological malignancies using the talents. Okay, they are trying to mutate the HLA molecules to make it as a allogenic CAR T cells. Okay, this is already in clinical trial. Okay, so so these are all the zinc finger talons works, but it takes a lot of time to get one pair of zinc finger nucleus. It takes at least 6 to 8 months. Okay, and another major disadvantage in terms of zinc finger nuclease, the zinc finger nuclease technology was transferred to a company called Sangamo Biosciences, the patent, and then the Sangamo Biosciences was controlling the patent, and they were selling it for very high price in early 2000. So that time itself it was like more than $5,000. So they didn't give it for the research actually. Okay, so that's where people couldn't able to afford, many people in the US labs itself. And then say example, if you are going in a road, if there is any agitation is going on, you always take a detour, look for some other route, right? So the scientists got frustrated with the zinc finger nuclease patent and the high price, and then they were looking for something else. That's where the CRISPR-Cas9 came into the picture. Okay, so CRISPR-Cas9, what is CRISPR-Cas9? Okay, so CRISPR-Cas9 is a natural bacterial immune system, okay, which is very similar to our restriction enzymes. Okay, so when virus infects the bacterial cell, so the viral DNA just comes out of the bacteriophage, and then it has the bacteria has a CRISPR array. Okay, so this CRISPR array takes up this viral DNA, the foreign DNA, the specific invader DNA, and puts into the CRISPR array sequence. Okay, so this is the CRISPR array. Once it enters into the bacteria, it takes small piece of this viral DNA and it incorporates into the CRISPR array. So then it uses the cells its own machinery, the transcription and translational machinery, and then the CRISPR array gets transcribed and it produces the pre-CRISPR RNA, and then the pre-CRISPR RNA gets further processed and then it makes into tracer RNA. Okay, so this cRNA forms a complex with the Cas9 nuclease. Okay, so when next time the same bacteriophage, the virus infects, it uses this memory, okay, this small sequence which took it from the same bacteriophage, right? So it goes and binds to that the bacteriophage or the virus, and then the CRISPR-Cas9 system gets activated. Cas9 makes the targeted double-stranded break. It chops the viral genome into small pieces, and then the viral genome gets degraded. So now, after this, Dr. Charpentier and the Doudna, they have simplified this system. Okay, so this is already existing naturally in the nature, but what they have done is they simplified. Okay, so this is the natural CRISPR system. It has 3 domains. One is cRNA domain, which gives this 20 base pair specificity to the target sequence, and then there is a tracer RNA, okay, which gives the stability to the cRNA, and then you have the Cas9 domain. Okay, this is a native system. Okay, what they have done, Dr. Doudna and Charpentier, they fused this cRNA and tracer RNA, and then they made as a synthetic guide RNA. Okay, so and then that simplifies the protocol. Okay, so then the Cas9 comes into the picture. Once the CRISPR goes into the guide RNA, goes and binds to the target locus, and then the guide RNA makes the targeted double-stranded break, and then it cuts into the genome. Okay, so what you can do with these gene editing nuclease? Okay, there are multiple applications, the biomedical applications, and then it's in multiple fields. Okay, so I'm just giving an example here in this cartoon. So if you imagine this is the cellular genome, okay, so you have a 5' to 3' and 3' to, this is the double-stranded DNA. Say example, if you want to create a specific mutation, say example in a CCR5 gene, chemokine receptor gene, okay, so to mutate the CCR5 gene, okay, so you can develop a zinc finger talon or CRISPR using molecular biology approach, and then you can deliver it into a cell. In this case, it's a T cell, right? CD4, CD8 T cells. So CD4 T cell. So then it goes and binds into the target locus here. So imagine this is the CCR5. It makes the targeted double-stranded break. So if you leave it just like that, so cells it has its own repair mechanism. Okay. So then it uses the sister chromatid and it gets joined. Okay. If it is not joined, then it will get degraded. So it has to get combine. It has to undergo the homologous recombination. So there is one process is called non-homologous adjoining. So when you make a cut, this process of non-homologous adjoining, when it is joining, it creates a small base pairs deletion, substitution, or long deletion as well. It is called indels. Okay, so that puts a error in the sequence. Okay, it may create a premature stop codon. Okay, if you have a gene, so it's ATG to TAA, okay, TAG or something like that. So because of this targeted double-stranded break, the stop codon comes in the exon 1 or exon 2 itself, wherever the gene editing nuclease targets. So then if you produces a premature stop codon, then the protein will not get produced. Okay, so it will be non-functional in the case of CCR5. Okay, so then the HIV can't enter into that. The other case, you can use a donor DNA which has your correct sequence. Okay, so in say example in the case of sickle cell anemia, right? So if you want to put a GAG sequence instead of GTG, so you can make this donor sequence with appropriate homologiome. Okay, so this is the correct sequence you want to insert it, and then this has the 5' homologiome, and then this portion is the 3' homologiome, left and right homologiome. Okay, once the targeted double-stranded break happens, it goes and binds to the target locus using these homologiomes, 5' and 3' homologiome, using cells its own repair mechanism. Okay, you are providing this externally. Okay, so either you can use donor DNA or single-stranded oligonucleotide. So then the gene correction happens, homology directed gene repair happens. Okay, the third application is, so if you want to produce a more enzyme, say in the case of Gaucher's disease, okay, glucocerebrosidase is an enzyme deficiency which causes the glucocerebrosidase, right? So now the enzyme replacement therapy is the only available therapy currently for these Gaucher's disease patients. That is even for the only for the splenomegaly and hepatomegaly cases. If the patient has the neurological effect, this also will not work. There is no therapy at all. There is no small molecule. There is no enzyme replacement therapy for the patients with Gaucher's disease who has the neurological defects. But if you want to make a more transgene for these, make the enzyme replacement therapy, you can insert a transgene, say example this GBA gene, which produces the glucocerebrosidase in the human genome. Okay, in our human genome, there are some regions are called safe harbor locus. Okay, one such safe harbor locus is the CCR5. Okay, as we all know, I will come to that. Why CCR5 locus? It is a safe harbor locus. You can insert the transgene into the safe harbor locus of CCR5. The second locus is adeno-associated viral locus. So even if you mutate those locus, it will not affect our cell growth and development. That's why it's called a safe harbor locus. So then you can put it into the cell, make more enzyme from that, and then you can infuse it into the patient, or you can take the patient's own macrophages and then put these transgene using gene editing nucleases, and then the macrophages will make more amount of glucocerebrosidase, so that will reverse the disease phenotype. Okay, what are the biomedical applications of this targeted genome engineering? One example is there a cure for HIV AIDS? Yes, there are already multiple clinical trials are going on. So as we all know that HIV uses two co-receptors. Okay, one is the CCR5, another one is CXCR4. The first line is the CCR5. Okay, so this is like a door here. Okay, so if you are entering into the home, you enter into a door, right? So the CCR for HIV, the CCR5 is like a door. Okay, so if you mutate this door, that is the CCR5 gene using gene editing nucleases, you are shutting this door. Okay, So you are making the CCR5 as a non-functional, okay? The door is closed, okay? So then the HIV can't able to enter into the human T cells, okay? So that is the basic concept here, okay? So HIV requires two core receptors, one is CCR5 and CXCR4 to enter into the human cells. But there are individuals, as we all know, that across the world there are tiny group of population, they have a mutation called delta 32. There is a 32 base pair deletion, natural deletion in their CCR5 gene. So if they have a biallelic mutation in both the alleles, they have the, if they have the mutation, they are permanently resistant to HIV. Okay, if they have one allele, while one allele has the mutation, the second allele is the wild type, they have 50% resistant to HIV. Okay, but these people live very normal and healthy life, not associated with any pathology. Okay, so one best example is a Berlin patient. There are multiple examples here. Now there is a London patient, you all might have seen in the newspaper and other news channels. This is very long back. It is, this patient is called Berlin patient. He had leukemia as well as HIV. Okay, so he undergone bone marrow transplantation. Okay, so fortunately he got a donor belongs to this CCR5 delta 32 mutation. Okay, so this is where the clinicians role is very, very important. So if they know that the CCR5 delta 32 mutation can cure the HIV, so you can look for the patient, though it is very rare mutation, but if we are lucky, we can get the donor, patient donor, HLA matched donor belongs to the CCR5 delta 32 mutation. So he undergone bone marrow transplantation, and then after this bone marrow transplantation, his viral load was very, very low, very, I think it was nil, almost like a nil, and for so many years, okay, for almost like 7-8 years. But unfortunately, he died because of the relapse due to the leukemia, not because of HIV. So this is one example. It shows that if you take the if you mutate the CCR5 gene, if you have the mutated CCR5 gene, you can completely cure the HIV. So now you can take this natural solution. You have a tool like gene editing nucleases. Take the patient's T cells, mutate the CCR5 gene, put it back into the patient so that the patient can get completely cured. It's a one-time cure approach. Okay, so this is what the thing. So you can take the patient's CD4 T cells or CD34 hematopoietic stem progenitor cells through apheresis, do genome editing, and then the CCR5 get edited and then put it back into the patient so that the patient will get cured. Okay. The second example I just want to give you, the sickle cell anemia and thalassemia. I'm not going into what is the pathology and other things because for this audience, so because you are all clinicians. So only one thing I want to emphasize here, currently hematopoietic stem transplantation is the only curative option available for Indian patients, okay, available to the Indian patients. Okay, so even though there is Bluebird Bio lentiviral based approach or the CRISPR therapeutics are there for US patients and the Europeans, but Indian patients we don't have the other curative approaches. So I just want to give a brief introduction about how these hemoglobin genes are developed, regulated during the development. Okay, so there are 5 genes in our, as you all know, that there are 5 genes in the beta globin cluster at chromosome 11. So all these 5 genes are regulated by a master regulator called locus control region, which is present upstream of the upstream of these genes, 5 genes. Okay, you have embryonic hemoglobin. During the embryonic stage, the locus control region forms a loop with the promoter region of the embryonic hemoglobin, and then it starts expressing, and then which takes care about the oxygen transport. Okay, so after first the 6 to 8 weeks of the gestation, this embryonic hemoglobin gets silenced, and then the loop gets changed into the promoter region of either HBG1 or HBG2, which is nothing but the fetal hemoglobin. Okay, so during the fetal stage, the fetal hemoglobin takes care about the oxygen transport, right? So the second switch happens after the birth, maybe within 6 months to 1 year. Okay, so both embryonic hemoglobin and the fetal hemoglobin gets silenced, and then the locus control region forms a loop with the promoter of the adult hemoglobin. Okay, so that's where the adult hemoglobin starts expressing at the adult stage almost like 99.9% of the people, we all express the adult hemoglobin, which is the HbA or HbB gene, right? So B protein. Okay, so but there are some individuals across the world, they still express, even at the adult stage, very high level of fetal hemoglobin, even at 90% fetal hemoglobin at the adult stage. That condition is called hereditary persistence of fetal hemoglobin. Why that happens? Okay, so that is because of the natural mutations. Okay, as I told earlier, so this during the adult stage, there are some negative regulators like BCL11A, okay, so which goes and binds to the promoter region of either HBG1 or HBG2, and then it suppresses or it silences it suppresses the fetal hemoglobin expression. So that's why the loop gets changed into a HBB and it starts expressing. Okay, so these are all the point mutations. So if there are some individuals across the world, they have point mutations in the promoter region of the fetal hemoglobin. If they have the point mutations, these negative regulators like BCL11A or ZBTB7A, these are the transcription factors, will not bind at the adult stage. Then those people, the switch doesn't happen at all. So that's why they express high level of fetal hemoglobin. The second type is called, there is a long deletion, okay, from after this the HBG1, it varies different ethnic groups. Okay, so hereditary persistence of fetal hemoglobin is a natural way to enhance the gamma globin. Okay, so people with the sickle mutation or thal mutation who co-inherit this the promoter mutations, they live very normal life without knowing they have sickle or thal mutation. Okay, so around 20 to 30% of, as you all very well know, that is sufficient to reverse the disease phenotype. Okay, so naturally occurring HbF inducing HbFH mutations reverse the clinical phenotype, and it is much more safer. Okay, the question is, can we able to make these mutations in the CD34 hematopoietic stem cells? Okay, so these are all the different mutations, non-deletional HbFH and deletional HbFH. You can see different ethnic groups have different mutations. These are all mostly single base pair substitutions. They all have high level of fetal hemoglobin. And then similarly, the Indians also have the tiny group. They have 50 kb deletion. They have high level of fetal hemoglobin. We have seen in multiple families in Odisha and Mumbai, they have high level of fetal hemoglobin across generations because of these HPFH3 mutation, which is a long deletion in the beta globin cluster, which completely deletes the HPB gene. Okay, so there is no cell growth and there is no problem in cell growth and development for these individuals who carry this HPFH3 mutation. So the question my students Pragya and Sangam, they asked is, can the developmentally silenced fetal hemoglobin gene be reactivated in patients' adult erythroid cells by juxtaposing HBG1 or G2 with the locus control region? That is the, instead of forming a loop with the adult gene, can we reverse back into the fetal hemoglobin? That's the question we asked using genome editing. Okay, so that is where there are multiple different approaches you can use for beta hemoglobin disorders. One is gene correction. You can use homology directed repair. But what happens here is the targeted double standard break happens throughout the cell cycle from G1 to M phase. Okay, but gene correction using homology directed repair happens only at the late S and G2 phase. Okay, so that's where the efficiency of the genome editing in terms of homology directed repair is less, very less. Okay, so especially in primary cells like quiescent cells, CD34 hematopoietic stem progenitor cells, the homology directed repair is very low. The gene correction is relatively less compared to the cell lines. Okay, but can you go around this? So can we can we mutate this BCL11A gene, which is a negative regulator? That's what this CRISPR therapeutics they have done. They have mutated the BCL11A gene. so that it will not bind at the promoter region of the fetal hemoglobin at the adult stage, so that you can reactivate the fetal hemoglobin at the adult stage. So what we are trying to do here is we are trying to create a beneficial mutation in patient CD34 hematopoietic stem cells using CRISPR-Cas9, so that it will be a one-stop solution for both sickle and thal. So you are basically reactivating the fetal hemoglobin, so that will be pathologically more acceptable. So already the nature has given a solution. So this is our hypothesis. So in the disease phenotype, you can see here, because of the mutation, you have low level of adult as well as fetal hemoglobin. That's where leads to the disease phenotype here. But if you can able to create these beneficial genotype in patient CD34 hematopoietic stem cells, you can change the loop from here to here into the fetal hemoglobin. So that way you can increase the fetal hemoglobin expression in the CD34 hematopoietic stem cells, so that you can reverse the disease phenotype. Okay. So one bottleneck here, as you all mostly the hematologist. So the major bottleneck here is the CD34 the frequency of CD34 hematopoietic stem cells in human peripheral blood in the circulation is very low, 0.01 to 0.1%. Getting enough CD34 cells for even R&D purpose is extremely difficult. Okay, this is a major challenge. So how to go around this? Okay, so in order to go around, so we asked the question, my student asked the question, can we generate pathophysiologically relevant erythroid progenitor lines for sickle cell and thalassemia? So what we have done here, so we have taken a erythroid progenitor line and created a created a sickle and thal mutation, and which these lines exactly mimics the CD34 hematopoietic stem progenitor cells derived from the patients. Okay, so this is a cell line, so that you can grow it into the laboratory condition. So if you want to test your hypothesis, or if you want to screen a small molecule library to reactivate the fetal hemoglobin, so you can use these cells. So these progenitor cells can be differentiated into erythroid progenitor cells, enucleated cells, so that can be used to test various hypotheses. That's what we have shown in this paper. This came in this we published last year in Nature Communications, and then we use these similar cells also for the for identifying some drug molecules against malaria as well. So this publication very recently, just last month, came in Nature Communication Biology. Okay, so so then the real question is, does the beneficial genotype ameliorate the sickle cell disease? Okay, so that is where, so we took 3 patients, sickle cell patients, SCD 1, 2, 3, and we used this CRISPR-Cas9 and created the beneficial genotype in CD34 hematopoietic stem cells. Okay, so what we have done here is we took backup cells. When you go for when the patients go for bone marrow transplantation, so we took these samples from Dr. Gaurav Karya from Apollo Hospital. So with he keeps backup these cells in case the patient's engraftment is poor, from the donor cells engraftment is poor, so that you can save the patients, right? So these cells, after 6 months, these are all to be discarded cells. So this was taken ethical approval and then used for the basic research purpose. So we took these cells and then we put the CRISPR-Cas9 into these cells, and then we first checked the genome editing efficiency, and then we achieved almost like 60% double-stranded knockout, generated the beneficial genotype, and then we differentiated them into enucleated cells. So you can see clearly, even after genome editing, the enucleation efficiency doesn't alter. Okay, so that is one good sign that the differentiation potential is not hampered because of gene editing. So the next question is whether this will increase the fetal hemoglobin expression. Okay, so in order to test that, there are some there is a cell called F cell. Okay, so which these F cells produces the fetal hemoglobin. Okay, these F cells there are specific markers are there. So we have used the F cell fetal hemoglobin antibody through the flow cytometry. We checked how much percentage of the fetal hemoglobin is produced in these edited cells. So you can see clearly that almost like 60% of the cells produces the fetal hemoglobin, which is almost like six-fold increase than the unedited counterpart. So The take-home message is generating HbFH genotype had no observable impact on the enucleation rate in edited cells. F cell population was significantly enhanced in all three patients compared to the unedited controls. Next question is, do HbFH edited erythroid cells rescue the sickling phenotype? Yes. You can see here after the genome editing, the HbF levels has gone up in all the three cases. I am just showing in one case here as a representative. So here you can clearly see the reversing of the sickle cell phenotype, that has the sickling levels has significantly gone down in these. So the HPLC measurement showed high fetal hemoglobin reactivation and clinically relevant HbF expression in all three patient-derived samples. Under hypoxia, all edited samples showed fewer sickle cells and sufficient HbF to prevent the polymerization. So the last part is, does the HbFH3 mediated induction of HbF ameliorate beta thalassemia phenotype? We have seen sickle cell, it reverses, yes. Whether it can also help in reversing the beta thalassemia phenotype. So in order to do that, so as you all know that in the beta thalassemia cases, the major problem with these patients is the reduced erythropoiesis. Okay, so these cases, the number of enucleated cells, the generation of high number of enucleated cells is very less. Okay, so when we knocked out, when we created this beneficial mutation, when we checked the enucleation efficiency compared to the unedited or mock treated one, you can see very clearly see that this erythropoiesis, the enucleation level has gone up significantly. So this itself shows that this beneficial mutation can reverse the disease phenotype. And then F cell population has also gone up in these all three patients, BT1, BT2 and BT3. So the delayed enucleation was observed in unedited samples due to the ineffective erythropoiesis, but it was rescued in HbFS3 edited samples. So does the HbFS3 reactivated HbF rescues the beta thalassemia phenotype? Yes. So the globin chain gets balanced, and then the ROS levels, the reactive oxygen species, which is very high in these patients, has significantly come down in all three patients. Okay. So the next important question is, once you edit these in vitro, is whether these cells can engraft. That is the most important part, right? So before going into the patients, we can use the immunocompromised mice. There are mice which is highly immunocompromised, no B cell, T cell, NK cells are present in these mice, very susceptible to infection. So we purchased these mice from the Jackson laboratories from USA. We edited these patient cells and created the HbFH3 beneficial genotype and then injected into the mice and then checked the engraftment potential for long term, 16 weeks. Okay. So, and then at the 16th week, we euthanized the mice, collected the bone marrow, thymus, spleen, and then tested the different things. Okay. So here is the engraftment. After different weeks, you can see 4 weeks, 8 weeks, 16 weeks. These are all no injection, unedited, and HbFS3 edited. So you can see clearly after genome editing in both sickle cell and thalassemia cases, the editing doesn't hamper the in vivo engraftment. Okay. So and then we have also checked the multi-lineage potential, whether it can able to differentiate in vivo into myeloid cells, T cells, B cells, macrophages, and other things. Okay, that also, which is very equivalent to non-edited and mock treated. And then finally, we also check whether even after putting it into the mice, whether it can able to retain the F cell population, and then it can able to bring down the sickle and ROS levels. Yes, which can able to bring down the ROS and sickle levels in both the cases in BT as well as SCD. So the long-term repopulating cells in mice retain the HbF editing at similar levels to the infused cells. HbFS3 edited HSPCs repopulate for long-term and produces the HbF cells. The efficient reactivation of HbF resulted in reduced levels of sickle and ROS in BT samples. So this is the take-home message. So what we have done is, we took the CRISPR-Cas9, we generated the beneficial mutation, and then this is the disease phenotype. And if you create a beneficial mutation, you can change the loop from the adult to the fetal hemoglobin, either HbG1 or HbG2. So this level, HbF level goes up, and HbS, the mutant level, goes down. Because of that, it can able to reduce the sickling and the restored globin chain balance, and it also brings down the ROS levels, and which also we have shown that the genome editing doesn't alter the genome integrity, and then we have achieved long-term engraftment and the multi-lineage potential. So this is the overall approach. If you want to take it to the clinic, so you can take the mobilized patient, you can mobilize it using GCSF or Plexi4 at the hospital, take the patient's cells through the apheresis, and then separate it using Clinimax Plus or Prodigy system. We can take it into the GMP facility, and then using the electroporation, you can deliver this, which is a non-viral approach. You deliver the CRISPR-Cas9 into the CD34 cells, and then do the battery of test, put it back into the patients after the conditioning, so that it will get engrafted. So this is the overall approach of the ex vivo gene therapy. So with this, I will stop here. So this work was mainly done by Pragya Gupta and Sangam Goswami. Both are very talented PhD students. Now Pragya is in AstraZeneca, Sweden. Sangam is in Virginia, US, University of Virginia in US. And then this work was supported by very talented other students from my lab, Vinod, Aditya, Nupur, and Mavish. So this work was done in collaboration with multiple hospitals, main support from Dr. Gaurav, and then Dr. Suman Jain from Thalassemia and Sickle Cell Society. So in collaboration with Dr. Sailaja Singh, Avinash Bajaj lab for the animal work. Okay, so with this I will stop here. I'll be happy to take any questions before going into the other session.
Yeah, thank you, Dr. Shiva. That's a fantastic talk and a brilliant disposition of the work that you're doing. I mean, what is happening on Indian soil, as uh, Dr. Kateva rightly said, that most people sitting in India are unaware of the work that goes on in our own country. We are always looking westwards to see what's going on. I mean, I'm a personal sort of experience with that. So I really appreciate what you have done. And I think all of the, 50 odd people over here, or maybe more, would have understood the depth of your work, which is no less than the sort of gene editing work which has been carried out abroad. And the depth to which you have gone to sort of make this work translational is really phenomenal. So, saying that, I would open this forum for discussion. So I think there are some questions. So Rajat Bhattacharya has asked a question that what he talks about is the Anygame recent paper where in vivo gene editing to cure CPS1 deficiency is sensational. But do you think that this can be generalized for widespread use?
Yeah. Yes, sir. Please go ahead. Yes, yes.
Yeah, he says it's very labor intensive, costly, and what is the future of in vivo gene editing for such diseases?
Yeah, this is a very fantastic question actually. So it will bring down the cost significantly because we no need to depend on the GMP facility and other things. That is where even in fact we are thinking in that direction only. In fact, we had a lot of discussions. Even Gaurav used to tell the same thing. To bring down the cost for the Indian patients, we need to go for the in vivo delivery. So the major bottleneck here is we don't have a specific receptor to target the CD34 hematopoietic stem cells. There are some receptors, say example c-KIT or CD90, CD47, but these receptors also expresses in other cells as well, liver or lungs. So you will have some kind of off-target effect as well. So so that's where this field is little bit stuck here, but this is the long term goal is, or everyone wants to develop in vivo delivery of this CRISPR-Cas9 in targeting the CD34 hematopoietic progenitor cells. There are many groups are working on it, targeting different things. There are 1 or 2 papers also came out, but still the specific receptor is still not exist for this targeting the CD34 hematopoietic stem cells. But that's where the field is moving actually. Yes.
So I think Deva Jyoti at IGIB is doing some good work in sickle cell field as well. I wish he was here today to join the discussion. It would have been fantastic because they have got some very innovative CRISPR-Cas9 system in place. Dr. Vinod was asking about what is the translational prospect of this work on BCL11A in terms of taking it to bedside for sickle and thal.
Yeah, so BCL11A, the CRISPR Therapeutics has already taken into the clinical trial, right? So it has been already approved. So here our approach is creating a beneficial mutation in the beta globin cluster, which is totally different than the BCL11A approach. So we are creating a beneficial genotype. So that's where we are trying to reactivate the fetal hemoglobin. So this has major advantage than the BCL11A here, because here we are also deleting the mutant hemoglobin gene. Okay, in BCL11A the mutant hemoglobin gene is not deleted. Okay, so here we are also deleting the mutant one, so that has double advantage than the BCL11A. So you are also bringing down the expression of the sickle hemoglobin drastically, along with the fetal hemoglobin. Okay, so that is the one major advantage with our approach.
Yeah, that's great. So see, the issue of taking it to the clinic does not rest with Dr. Ramalingam alone. So
Right.
this translational work has to have clinicians to have better understanding of the interface of the lab and the clinics. So how we take it to the lab. I mean, Gaurav in that respect has done a fantastic work. Dr. Gaurav Khare, I mean, over here. I mean, he's been sort of a beacon for all the clinicians, that how a clinician can take interest in basic research and lead the way to translational work. So with this, I think we
Yes, just
can.
I want to add up, sir. So,
uh,
so right now we have done the both in vitro and in vivo preclinical studies. So there are some toxicology studies is pending. So if we want to take it to the clinical trial, yes, you rightly said, we need to have a right kind of clinical collaboration for this. So we are working towards that. So it may take some time. So you need to, because you need to get the GMP grade material. So all these things now it was done in research grade reagents, right? So when you want to go into the translational aspect, so we have to test this one round with the GMP grade reagents before taking it into the clinical trial or anything. And you rightly said Dr. Gaurav is phenomenal. So he is pushing all of us for the Indian patients. So for the benefit of the Indian patients, for both sickle, thal, and as well as for other malignancies as well. Right.
So,
so I
thank
think,
you. I think we are
yeah,
running
I
late.
think we can go on the panel discussion.
Yeah. Thank you, Dr. Suparno and Dr. Shiva, for giving this
Thank you.
excellent session to all of us.